let's talk about synapses. So, to start off, it is important to know what we're talking about. So, here is my really great drawing of a synapse. You can see the synaptic cleft, that's just the space between two neurons, and you can also see the presynaptic membrane and the postsynaptic membrane. These are pretty aptly named, as the presynaptic membrane is prior to the synaptic cleft, pre, and it releases the neurotransmitters. And the postsynaptic membrane is after the synaptic cleft, post, and it receives the neurotransmitters. It's also important to note the synaptic vesicles, which are where neurons store small and frequently used neurotransmitters, and to know about storage granules, which are for larger neurotransmitters. So, how does it happen? Well, there are a couple steps to synaptic transmission. It all kicks off when an action potential travels down an axon and into the presynaptic terminal. That's near the presynaptic membrane that we talked about earlier. Now, the resulting depolarization enables calcium voltage gated channels to open and for calcium ions to enter the terminal. And when these calcium ions enter, they cause the synaptic vesicles to then fuse with the presynaptic membrane, therefore pushing their neurotransmitters out into the release zone. And now that the neurotransmitters are out in the release zone, they diffuse across the synaptic cleft and attach to the receptors on the postsynaptic membrane, where they make their impact on that neuron. Now, we could go further with that, but that's not what we're talking about right now. We're talking about synapses. So, back to these neurotransmitters, they then separate from the receptors, after which they might diffuse away, or they might be transported back into the presynaptic neuron by transporter proteins in order to be recycled, and that process is called reuptake, or they might be broken down by enzymes in the synaptic cleft, and that process is called enzymatic deactivation. Okay, so switching gears a little bit, let's talk more action potentials. Some important terms to know here are excitation and inhibition. In neuron speak, excitation is synonymous with depolarization, and inhibition is synonymous with hyperpolarization. Remember, these are all about the electrical charge and membrane potential. So, where the neuron's charge is in relation to the threshold for an action potential. Now, excitatory postsynaptic potentials, EPSPs, are graded depolarizations. So, sodium enters the cell and the membrane potential climbs, but not high enough for an action potential. Remember, it's graded. Now, inhibitory postsynaptic potentials, IPSPs, are graded hyperpolarizations. So, chlorine exits the cell and the membrane potential is brought further away from the threshold for an action potential. And these two things are important because these EPSPs and IPSPs, they impact how easily and therefore how often a neuron can produce an action potential because they impact how far the membrane potential has to climb before reaching that threshold. So, speaking of action potentials, there are some pretty cool ways to get there that I think we should talk about. So, the first of these is temporal summation, or summation over time. The combination of subthreshold excitations, if close enough together in time, can produce an action potential. And you can think of this as stimulation to one point twice. And the second type of summation is spatial summation, or summation over space. In this case, the combination of subthreshold excitations, if close enough together in space, can also produce an action potential. And you could think of this as stimulation in 2 points at once. And for both of these types of summation, EPSPs are a very important part of these, and they make it more likely for summation to reach an action potential. And on the flip side, on the other hand, IPSPs make this less likely. So these two functions, EPSPs and IPSPs, they help regulate neurons' action potential frequencies, and they make it more or less likely for a message to be passed on. So this was my quick little review session. I hope that this was helpful and relatively concise. I do want to source this last photo that I have on this slide, as I did not create this last one. So that is all, and thank you.